Good afternoon. My name is Mogens Vyberg. I am the director of the NordiQC program, and I have been kindly invited by Merck to give this presentation on the evaluation and optimization of laboratory-developed immunohistochemistry 22C3 assay for PD-L1. The study has been carried out by the NordiQC staff, guided by Søren Nielsen, our scientist and manager. First, a few words about the NordiQC quality controls or quality assurance scheme. We are an international organization founded in 2003, independent, scientific and not-for-profit organization based at the Institute of Pathology, Aalborg University in Denmark. We run for a general module with 3 runs per year with 15 to 18 different marker challenges. We have a breast cancer immuno module with 2 runs per year, a HER2-ish inside of hybridization module, and we have also recently opened a companion module as a pilot, and from the autumn this year I suppose it will be open to all participants in NordiQC. This is a screenshot from our website. It's open for everybody as regards the general information. The address is www.nordiqc.org, where you can see the menus and the photos. You can access the assessment schemes during 2017, all the markers included here for the general module, breast cancer module, and inside hybridization module. You can see all the dates where we specify when the protocols are to be submitted and when the slides are shipped and so on. For participants, they can log into our site and see what is going on. Is their protocols registered? Did they receive the slides? And did we receive the slides? And so on. The protocols requested for all assessments are available for the participants, and they can at any time modify the protocol if they, for example, include a new batch in the assessments. We send out the test material based on multi-tissue formalin-fixed paraffin-embedded blocks. All tissues are fixed according to the ASCO-CAP guidelines when relevant, and we include in these tissue blocks normal and clinically relevant tumor tissues, focusing at different levels of antigen expression, high, moderate, low and non-expressions. We make 2 unstained slides for each marker sent to the participants, and 1 should be sent for return for central assessment. Test material is composed, as I said, of tissues with varying expression. Here 1 example on BCL6 test where we have included 2 tonsils with varying expression or varying fixation, 2 lymphomas with varying expression, or 3 lymphomas actually. And you can see here on the photo how we can identify areas with high expression, the follicular germ cells. We have the low expressing cells, the epithelium, here the squamous epithelium in the tonsil, and the small lymphocytes are non-expressing cells. We perform detailed description after each of these tests, and where you can go to the website and see what are the requirements for optimal staining reactions and how did the different antibodies and protocols perform. And to the participants we sent, particularly when assessments were not optimal, particularly when they are borderline or even poor, we detail the causes and we suggest improvement. We started in 2003, as mentioned, with only the Nordic countries, but have had the scheme open, and you can see in 2009 we had about 200 participants, and it has increased to about 700 this year. Going through about 20,000 slides in the general module, we have seen that approximately 1/3 are optimal, 1/3 are good, and the last third are either insufficient, either borderline or poor. we call them insufficient. And of these, about 90% are too weak or false negative, while 10% are overstained or giving false positive reactions. For the breast cancer module, the predictive markers, we have assessed about 9,000 slides so far, and 1 out of 5 have been insufficient for diagnostic purposes. We have, for 9,000 slides where we found insufficiency, identified the major causes, namely that about 17% of the slides are insufficient due to less successful antibodies or ready-to-use products. In 20% of the cases, we have seen inappropriate antibody dilution. 27% of the cases, we have seen inappropriate epitope retrieval. In 19% of the cases, inappropriate detection kit as a major cause. And in 17% other causes like endogenous biotin reaction or drying out phenomena or technical errors of any kind. Many of our results have not only been published on our website, but also in peer-reviewed publications, mainly in the applied immunohistochemistry and morphological pharmacology. And we have recently published a series of papers on standardization of positive controls and negative controls, as well as a series called Evolution of Quality Assurance for Clinical Immunohistochemistry in the area of precision medicine, in 4 parts in applied immunohistochemistry. Now, for a few examples, we have here 2 serial sections stained for estrogen receptor by 2 laboratories, A and B. This is the ductal carcinoma, and both labs seem to give a very nice staining reaction. But these cells are high-expressing cells. There's a lot of estrogen receptor protein present, and we don't know what happens if we have tumors with a low expression. But you can see here, to the left in the lower row, lab A still will be able to stain cells with a low expression, but in the right corner, lower right corner, actually lab B has failed. It's a false negative. This would not be discovered unless there was a very good control as well. And you can see here we recommend the uterine cervix as a control tissue. And to the left, the upper row, you can see that the basal cells are weakly stained, but these are, as it appears, the best cells for identification of a proper protocol. You can see the basal cells here to the right are virtually negative. False positives rarely occur with estrogen receptor, but we have seen that. In this case, you can see in the lower left corner, with 3 clones, especially the very sensitive clone SP1 in 225 labs, there's absolutely no estrogen receptor protein in these tumor cells, but with clone 6F11 in 15 out of 37 laboratories, actually you have a faint homogeneous staining reaction that we must consider to be false positive for technical reasons that are only partly clear for us. When we started, only 45% of the laboratories actually passed the test. It has improved quite fast according to our directions, and then we opened a new module. It went down. You can see slowly increase again. And then in this period we had a very large number of new laboratories participating, and the percentage of tests passed fell again. But now it's very close to 90%, which is okay. For HER2, you can see here the upper row giving optimal staining reactions for an amplified 3+ tumor, an amplified 2+ tumor, unamplified 2+ tumor, and an unamplified tumor giving no reaction. In the lower row, you can see the problems that frequently occur if the labs fail. It's still a very strong staining for the high-expressing tumor, but for this 2+ tumor with an amplification that should be a 2+, actually, the weak reaction indicates that now it's a 1+, and this patient would not be flexed to FISH to have further analysis, and then would lose the chance of getting treated. And for another case here with overstaining, still 3+ is 3+, and this amplified 2+, 2 or 3+, it would not matter. But here you can see it's 3+ staining in an unamplified 2+ tumor. So this patient would have a treatment of no value. During a number of runs, you can see how some of the FDA accepted approved tests have performed quite nicely, particularly the pathway system. But you can also see that laboratory developed tests have all often failed. Actually, we started with only 20% passing. It has been increasing, maybe due to the recommendations. So now it's approaching the level, but still it's below what is acceptable. It has some consequences. Actually, we made a study together with Roche about the consequences of having laboratory developed tests, and as you can see here, actually the costs or the loss of patients is tremendous. Per year it is normalized to the American breast cancer population in order to make some calculations on the costs, and if you just ignore in this case the patient's situation, actually the economical consequences are quite dramatic. So actually the conclusion of this study was that for each $1 saved by the pathology lab by usage of cheaper reagents, the healthcare system is ultimately burdened with $6. The consequences of participating in this external quality assessment scheme may be illustrated with this table. You can see for estrogen receptor, the pass rate, optimal and good, is quite high for the old participants compared to the new participants. Actually, the difference between the new and the old ones are close to 20%. So it seems to be quite helpful to participate in an external quality assessment scheme. Yet, 1/3 of all immunostains produced by NordiQC participants are still insufficient after we've been working for almost 15 years. There may be several explanations. New laboratories coming in all the time, new antibodies, techniques, platforms, and of course also increasing demands. What could be acceptable for 10 or 15 years would not be accepted today. My question could be how many immunostains produced by labs not participating in an external quality assessment scheme are insufficient. Nobody knows, but it might well be about 1 out of 3. And how many scientific publications are actually based on insufficient immunostains? That may also be quite high. And the last question is, what are the consequences for the patients? Well, you got an idea from the HER2 and estrogen receptor runs. So the conclusion is that external quality assurance provides objective evidence of a laboratory performance, identifies methodological errors, and provides directions for improvement and controls. And the results from our studies in NordiQC indicate that improvement of immunohistochemistry is strongly needed, and external quality assessment schemes, industry and key opinion leaders must align in describing the requirements for optimal immune performance. Now, going to the PD-L1 issues. The first question is how to implement a new biomarker such as PD-L1 for diagnostic purposes. How to focus on the requirements and challenges for the calibration and validation of these assays. And then we'll talk about the preliminary data on in-house validation for 22C3 as laboratory developed tests for PD-L1. Here you can see this is not PD-L1, but it's EpCAM, epithelial cell adhesion molecule, illustrated to give you an idea of the issues of biomarker calibration and validation. In the upper row, you can see the normal colon mucosa stained with 3 different protocols, and you could be satisfied with all 3 stains. And if you had the staining to the right in protocol using protocol 3, you will never discover actually that something might be improved because virtually all cells are nicely stained. If you look at a colon adenocarcinoma, which is often a high expressor of EpCAM, you can see very strong staining in all 3 cases. But looking at renal cell, clear cell carcinoma actually, that's typically a low expressor, you can see that only protocol 1 actually passed the test with a very nice membrane staining. Very faint reaction is seen with protocol 2, but no staining at all with protocol 3. Using another control instead of colon mucosa or even the colon carcinoma, look at the normal kidney here, where you have the strong staining of the distal tubules. You have very nice but weak staining of the proximal tubules, and also the Bowman capsule is very weakly stained, but it's distinctly reacting here for ECCAM. But if you dilute the antibody or your retrieval is insufficient, actually you will lose a lot of information. And this is quite parallel to what you can see with PD-L1 staining reactions. So from antibody to diagnostic, predictive and prognostic immunohistochemical biomarkers. Going first for the technical calibration, we must identify the best practice protocol. We must have the antibody calibrated, identify the signal to noise ratio, be sure about the reproducibility, consistency of the assay, and so on. We have to evaluate the impact of pre-analytical parameters like the tissue quality and some variables in the tissue processing. We have to identify the positive and negative controls that are both best apt for these assays. And we have to look at the interpretation guidelines in comparison with the technical calibration to end up with defining the sensitivity, the specificity, the accuracy and precision of the study. One question is, what is the intended purpose, or what are the intended purposes of your test? Here, for example, we have an ALK staining using a lung adenocarcinoma protocol to the left. You can see almost an over staining of this large cell anaplastic lymphoma, but you can see a very nice, strongly stained lung adenocarcinoma. To the right, you have the lymphoma protocol, and still a very nice staining for ALK in the lymphoma, but the lung cancer is false negative. So you have to be sure about which protocol is used and for which purpose. Here for the PD-L1 immunohistochemistry approval, you can see this table that we have from Andreas Scheel, produced in the German ring trials, listed for melanomas, non-small cell lung cancer, Hodgkin's lymphoma, renal cell carcinoma, urothelial carcinoma, and others. And you can see that today actually only the use of 22C3 assay for non-small cell lung cancer is mandatory, both for second line and now also for first line therapy. But most likely the test today, being optional, will sooner or later actually be moved to mandatory tests. Here you can see how 4 cancers give very different reactions from 4 assays produced each from one company. And you can see here with the very strongly expressing adenocarcinoma, the difference is very small. But for tumors with low expression, you can see increasing variation, and there may also be some issues about which cells are actually staining. Are you staining the tumor cells, or are they infiltrating histiocytes or other host cells? This is a table showing how the cutoffs are defined with the various assays, and some of these cutoffs are clinical relevant and others are not right now for the mandatory tests. When we score PD-L1, we focus on the percentage of the carcinoma cells with any membrane reaction for PD-L1, and in some assays also for the histiocytes, but not when we're talking about the non-small cell lung cancer. And eligible for treatment, depending on the first and second line, are 50% and 1% respectively. There are 4 assays and only one specific assay per drug. And you can see here listed in the upper table how these closed assays that are defined by the companies based on clinical trials, so that each of these 4 clones are linked to one specific drug treatment. While when it comes to the laboratory developed assays, we have a number of clones, 4 just listed here. They're sold by different vendors. They can principally be used on all platforms, but they are laboratory developed assays, and there's no drug actually related to these assays. We can see here how you can use the 22C3 clone related to the pembrolizumab drug as a ready-to-use product. No validation is needed before you start, but you of course need always to verify that the sting reaction has performed as expected using proper control tissue, for example cell lines as illustrated here in the bottom of the slide. But how to set up your assay if a proper immunohistochemical platform is not available, because about 80% of all labs will not stock the autostainer AS48 link. So you have to do something else, and this is why it is extremely important that a laboratory developed kit is calibrated in a way so it corresponds very precisely with the validated assays. Otherwise you will not be able to actually transfer the information. And you can see here on this slide how you have here a known risk to the patient for this assay 22C3 based on this drug pembrolizumab. But if you try to use pembrolizumab based on other assays like here, you have to obtain some new information about the clinical outcome, the risk to the patients, and even worse for the others. So there's an element of uncertainty in your setups here. So the validation of alternative PD-L1 immunoassays must be performed using the primary clinically validated assays as the reference. And as a minimum, it has been required by the ASCO guidelines that 80 to 100 samples should be used, and giving a 95% or higher concordance as regards positive and negative reactions based on the cutoff levels mentioned. There are some studies indicating here how it goes. This is from one of the clinical studies where it's comparing of the 4 assays, and you can see here that when 22C3 is compared with 28.8, for example, you have a cutoff of 95% concordance, but with SP142 and 263, actually the concordance is much lower, and this is not acceptable. In the German ring trial with 15 cases of non-small cell lung cancer and 9 observers, you can see here in this harmonization study, and these tables are not easy to interpret on the screen, but you'll be able to download the webinar. But the concordance have changed from 72 to 56 to 53, and in one case to just 41%, and this is simply not good enough. With these cutoffs, 1% and 50%, actually you can see that only 28.8 actually had a proper concordance, both with 1% and 50%. This is giving you an impression also from the same study how different the assays can illustrate the PD-L1 from a rather weak membrane to a very strong membrane staining, and of course it will also mean that tumor cells with a very low expression of PD-L1 will be negative with one assay but positive with another assay. Here you have one of the controls that we recommend for calibrating your assay. Tonsil is useful because you can identify the crypt epithelial cells that should give a stain reaction in the level of what's shown here. And actually this is the same tonsil series sections stained for PD-L1 with 3 different assays. But you can see, looking at the histiocytes in germinal centers here, there's a marked difference that comes up. This is squamous cell lung carcinoma with the same reaction, there's a strong reaction and much more than 50% of the cells in all 3 assays. And here you have another case where actually there's some difference. It's a weak expressor. You can see to the right, it's definitely about 50% of the cells being positive, while in the same field you can see with 22C3 it will probably be below 50%. So this is another illustration of this issue when you have to compare the different assays, and you can see with the comparison of 28/8 and 22C3, you can still have a very high cutoff, but with the others it is pretty much lower in both the so-called blueprint assay and the SHIELD study. So the problem is now how to transfer the observations from these different assays to laboratory developed protocols where we compare and really strictly try to calibrate laboratory developed protocols to make it comparable, so that you could be able to use 22C3 without having a DAGO autostainer. The study design that we carried out actually was based on 20 cases in a test set, a training set, and then 75 in a validation calibration set, where we had the 22C3 as a kit SK006 as a reference, and then we calibrated laboratory developed protocols on Bond, Omnis, and Ultra, and identified the concordance using these systems with the different cutoff values, 1% and 50%. This is how we composed the TMAs used for calibration, for training and for validation, with 22C3 as the golden reference, just to show you how even this small magnification you can see the varying intensity of the tumors. When we look at the protocol for the DAGO AS48 link stainer based on the 22C3 assay, we have basically a protocol with a high level of analytical sensitivity, has a relatively efficient heat-induced epitope retrieval, 20 minutes. It's a low pH buffer using the PT link. It's a 3-step polymer-based detection kit applied and prolonged incubation times. A lot of 3 steps, 3 minutes in each of them. The standard is somewhat lower, as you can see, and enhancement with copper sulfate after the DAB chromogen. The protocol for the Omnis, another DAGO stainer, is listed here. The protocol settings that we tried are given in the lines below, and you can see here how we tried various dilutions, various heat-induced epitope retrieval time, with the incubation time for the primary antibody, incubation time in linker and polymer, but kept the normal DAGO DAB as the chromogen. And the yellow text is indicating what we end up with to identify the protocol that gave the result closest to the results from the AS48 link. And here you can see illustrated the upper left photo here is a tonsil where you can see the staining reaction for the crypt epithelium and for the histiocytes in the germinal centers. And you can see for omnis with various settings where we choose the one in the lower right corner that we consider to be the one giving the closest reaction compared to AS48. Here we have a lung cancer illustrating virtually the same situation where we picked the one in the lower right corner. And here with another lung squamous carcinoma. And here with the third one, as you can see, very weak expression of the membranes. And you can see if we have used the other setups, actually there's a difference in the number of cells with a definite membrane reaction. So this is the one actually that gave the closest results. This is another lung cancer, and you can see approximately the same situation, that the photo up here in the upper left corner is most comparable to the one in the lower right corner. Now for the bond LICA, you can see again we have listed the protocol settings that we have tested, and we have marked in yellow the settings that should be used in order to have the best performance, the most comparable performance with the bond stainer. And similarly for the ultra benchmark Ventana, again marked in yellow the settings that we selected that could give the best staining reaction compared to 22-3 on the Dago 48. Selecting these setups, we finally end up in creating this test for validation, where we compared with all the 95 tumors in total, and as well as the controls, compared the staining reactions on the 4 stainers. And we found actually a very nice performance with 100% concordance for the 3 first ones, while for bond actually one of the positive more than 50 moved down to below 50 but above 1, and one of the tumors that should have been more than 1, it became negative. So the concordance rate was here 97%. Listing here with this graph the scores for each of the tumors, where we looked at the proportion score, not only at the cutoff values defined by the assays for 1 and 50%, but actually used at the each 10% level. You can see here that there's a very close relation, but a tendency for the staining reactions on the bond to be a little bit lower. And here you have some examples. And this squamous cell lung carcinoma, you can see that the staining reaction is virtually negative for tumors, because what you see as a positive staining actually are the histiocytes, and that is another challenge actually, to be able to discriminate between the histiocytes and the tumor cells, but it is not the point in this presentation. Here we have on the 4 platforms the AS48 Dago upper left corner, the Omnis upper right corner, the Ultra lower left corner, and the bond in the lower right corner. They give all a cutoff value about 1% and below 50. Here you have one where we have in all 4 cases also expression in more than 50% of the cells. Here another case where we have more than 50% in all of them. Here we have a further one still with more than 50% of the cells being positive. But as you can see here in the lower right corner, actually the membranes are getting pretty weak, and you have to go to a high magnification in order to be sure that you have actually a staining reaction. Here you have one other case with more than 1% in all of the 4 fields. Here with another, again more than 1%, but it's less than 50%. Again you can see, but with bond it's a little bit more difficult to identify the membranes. And here actually this is one of the cases where we consider the tumor on the bond stainer to be negative, because what you can see here mostly are the histocytes, while in the other 3 cases you can better identify the tumor cells giving this membrane reaction. This is a squamous cell carcinoma where the 3 of the assays actually gave a staining reaction in more than 50% of the cases, while on the bond it was about 1%, but it was less than 10%. But still, I should emphasize that the concordance rate became about the 95% expected. The conclusions from these studies are that using the 22C3, the SK006 on DAGO as a reference, it is possible to identify the best practice immunohistochemical protocol to provide concordant results for laboratory developed tests across the platforms. And that was the major task of our study. We could see that the concordance on the Omnis and Ultra was 100% and on the bond 97%, using the clinically relevant tumor proportionate scores. And still, to emphasize the differences for the absolute tumor proportionate scores was observed, and this is how it is. We also could see that we found a superior concordance of PD-L1 status for pembrolizumab treatment in non-small cell lung cancers compared to the SP263 immunoassay shown by a blueprint phase study and the PD-L1 harmonization publication by Dr. Scheel and co-workers. So that's something pointing towards the use of laboratory developed tests in order to have this calibration. But there's some drawbacks as well. I'll come back to that. We can see that access to and exchange of TMA material for confirmation of the immunotest accuracy is important in order to see the performance in different laboratories and to obtain evidence of the immunohistochemical method concordance. And it's particularly important also to map the so-called PD-L1 metrics. This means that what are the proportion of positive and negative cases in each laboratory. Another conclusion is that it's due to the complexity of validating the immunoassays combined with potential lot-to-lot variation in immunostains. We recommend basically to use the CDX or PharmDX assays for PD-L1, which conflicts a little bit with the recommendation on using laboratory developed protocols. The NordiQC data and publications have concluded that laboratory developed tests, however, generally are less reproducible and less cost effective than the assays for similar class 2 biomarkers, for example, the HER2 assay that I mentioned before. So, using appropriate PD-L1 positive and negative controls are important to monitor the consistency, the precision of the PD-L1 immunoassays. there are no perfect controls for PD-L1. Hopefully in the future the cell lines will give us better possibilities, but it is now recommended to use the tonsils, as mentioned a couple of times, and also to include tumors. For example, one that is totally negative for PD-L1, what is have a low expression for PD-L1, and one with a high expression of PD-L1. The perspectives just included here in the end, because we have not been able to study the staining reactions using image analysis, but several companies are actually presenting systems that are promising and seems to be quite important in the future. This is a poster from Flagship Bioscience that you can download. This is another from PerkinElmer, where they focus on the scoring of various phenotypes based also on the relationship between the PD-1 and PD-L1 positive stains in such a multiplex system. And here another example where Definiens and Merck have illustrated how you can basically by multiplexing have segmentation of the tumors, the PD-L1 and PD-1, and then make analysis on the proximity, and maybe in the future that will be very helpful for our diagnosis. So that is the end of the presentation, and so far we have not received any questions, but you are very welcome to send emails as I suggested, and then we will answer them afterwards. Thank you.